Hello, let's discuss this question. It's a question on osmosis. It's challenging question to some students, yet it's a very simple question. The diagram below shows an investigation of a strip of tradescantia stem, which were cut lengthwise from the main stem and then placed in distilled water and salt concentration or salt solution for about 45 minutes. And you can check the setup A, it was placed in distilled water, setup B in salt solution. Which are your expected results? We're saying this osmosis process. So the results in B is what? And we can check at B, your salt solution. Let's explain the observation. And the observation at this point, when you check this one, it's about the slit. What will happen to the slit? The slit will do what? It's about the curvature, not curvature, sorry, about the slit. What happened to this slit? The slit will close. So we are speaking about what? Slits close. Why does the slit close? The main reason for that is because the solution is hypertonic. Hence, this plant, cell sap, will lose water. Let's go to the accounting for your observation. And when you are accounting for your observation, we require three things here. Solution, number one. You speak about the process and your observation. So the first thing, you have to relate the solution. And the solution, yes, salt solution is hypertonic. So we are speaking about salt solution is hypertonic to those cells of the cortical cells. You get your first mark. The second mark, we are speaking about the process. So what happens? The hypertonic solution will exert high osmotic pressure drawing water through the semi-permeable membrane from cells above the cortical cells by osmosis. You get your second mark. And for the second mark to be there, this process should be mentioned by osmosis, leading to what? Leading to the slit closing. You get your third mark. And this is what I've explained. I've done a mistake. So this one should be B, not A. Sorry. It's about explanation for the B. I'll explain to you when we're explaining the observation at A. Then now, let's explain how the chemical affect the uptake of the mineral salts. You check the chemical cyanide. That is, you should understand first that is what? Enzyme. You are speaking about a chemical. So which is a what? Enzyme inhibitor. That is the first thing that you should know. We are saying cyanide. Before you write, you should know that it's a what? Enzyme inhibitor. Then you should know what is being affected. Respiration. Then the last one, you speak about the absorption. So the last answer should be on absorption. So let's now discuss or write about those three things that are required. So we are speaking about what? Cyanide is a chemical what? Inhibitor, which binds on active site of enzyme, you get your first mark. When it binds, thus affecting respiration unless or no energy is released. And because of this, what happens? Thus, low or no uptake of the ions. It can qualify to be 3, but because it's marked as 2, this will be our 2 marking points. But if it would be 3, this one would be another mark. So we are speaking about what? Explanation, how the chemical will affect the uptake of the mineral salts. Thank you very much.